The overall goal of this protocol is to describe the basic procedures to work with HEI-OC1 cells, which can be used to study the effects of pharmacological drugs on auditory cells, and assist in the functional characterization of the motor protein prestin.
HEI-OC1 cells are used by many researchers around the world in different experiments with different goals and using different techniques. Hopefully, the procedures described here will provide a common base to make these studies comparable. Of course, these experimental procedures may be further improved, but the differences with those described here could be key to understand and explain possibly contradictory results.
In this demonstration, we will describe how to work with HEI-OC1 cells, starting from a frozen aliquot to having samples ready to perform different studies with widely available techniques.
To begin this procedure, remove a vial of cryopreserved HEI-OC1 cells from liquid nitrogen and place it in a water bath at 37 degrees Celsius. Submerge only the lower half of the vial and allow it to thaw until only a small amount of ice remains in the vial. In a biological safety cabinet, wipe the outside of the vial with 70% alcohol. Then, pipette the cells from the vial and deliver the entire volume drop by drop slowly into a 15 milliliter conical tube containing 10 milliliters of pre-warmed growth medium, such as DMEM supplemented with 10% FBS. Next, remove the DMSO by centrifuging the tube for 5 minutes.